Hello everyone, so this is the GCSE Combined Science Trilogy paper, Biology Paper 2, that was sat by pupils in June 2018. Now I'm going to go through this and model how I would have tackled it. Remember, in the description of this video there's a link to the Markscheme and the blank paper, and there are also links to revision videos question by question, so you can see if there's any particular areas that you need to do some revision on. So, question number 1 then. Every year, scientists have recorded the date when migrating birds arrived at summer breeding grounds in the UK. The records show that for every 1 degree increase in mean global temperatures, the birds arrived 1 day later. What will the birds be competing for when they arrive at the UK breeding grounds? So we're on about competition. What do animals compete for? Well, the birds, they would produce eggs, so they wouldn't be competing for them. They would be competing for food, because that's what they want. They can't really compete for light. Mates is something they would also be competing for, because they're at the breeding grounds. And then oxygen, well, there's plenty of that in the air, so they're not competing for it. So the two things that they compete for are food and mates. Question number 2. Birds that arrive early might survive better than birds that arrive later. Suggest a reason why. Well, it might be because there's more food available, there's better shelter, there are more mates. Any logical answer, okay, it's only a suggest question and it's 1 mark. Any logical answer about why birds might survive better would get the mark, but try and link it to the things that animals compete for. Question 1.3. Global temperatures are increasing every year. This is because of an increase in greenhouse gases in the atmosphere. Name 1 greenhouse gas. Well, the 2 main ones are carbon dioxide and methane. Carbon dioxide, CO2, comes from burning fossil fuels. Methane, CH4, comes from agriculture, cattle rearing, etc. Question 1.4. Global warming affects the migration of animals. Give 1 effect of global warming. So you've got several options here, but the key things are rising sea levels or icebergs melting. Now I would say that is 1 mark, okay, don't say it is 2 separate things, because obviously the icebergs melting cause sea level rise. Then you've also got extreme weather, okay, then you've also got habitat loss. Okay, so some areas of the world are changing so much that they're no longer suitable for the animals living there. So any of them would give you the 1 mark. Which 2 human activities cause global warming? Okay, so we said burning fossil fuels. Well, that would definitely be it, because that produces carbon dioxide. Eating vegetables, well, that wouldn't release any greenhouse gases. Farming cows, as we said, produces methane, so that is your other greenhouse gas. Turning off lights, well, that wouldn't use energy, so therefore that's not burning any fossil fuel, so it's not that. And using too much water wouldn't release more carbon dioxide. Question 1.6. Which gas in the atmosphere causes acid rain? So we know CO2 and CH4 make global warming and climate change, but which one causes acid rain? Well, it is sulfur dioxide. Oxygen, obviously, is the gas that we need, so it's not that. Ozone protects us from harmful UV rays in the atmosphere. And carbon monoxide, that is produced from incomplete combustion. When you burn a fuel and not enough oxygen, instead of making CO2, you make CO. That binds with our blood and basically stops the oxygen getting round our body. Question 2 then. The genetic material in cells is made of DNA. Which 2 of the following describe the structure of DNA? Well, you should know that DNA is made of two strands coiled separately in different directions, and we call this shape the double helix. Okay, so the shape of it is a double helix, and then you've got two things you need to select. So we need one more from this list of four. Well, it's definitely not a single strand because we've got two strands coiling around. It's not a protein, okay, because DNA is made of building blocks, but it's not a protein itself. It's not a monomer because a monomer is talking about one building block together. It is actually a polymer because DNA is made of loads of different building blocks attached together to make DNA double helix. Okay, so it is a polymer and a double helix. Question 2.2, complete the sentences using the word from the box. A small section of DNA which codes for a protein is called a gene. Okay, a gene is a short section of DNA that makes and codes a protein. Okay, so all your DNA does is effectively tell your cells how to make specific proteins, which your body then uses. All the genetic material in an organism, well, it's not called a clone, it's not called a disorder, it's not called a mutation, therefore it must be called a genome. A genome is one complete set of your DNA, okay? And most cells have one complete set of your DNA. A mutation is the change in the DNA, okay? For some reason, your genes might have mutated ever so slightly. There might be when it copies itself, there might be a mistake when it copies, so therefore you get a mutation. Gametes, sex cells, contain around half the DNA compared to our normal body cells. Give the names of the two types of gametes, and your key thing here is sex cells. So, what are the two sex cells in humans? Well, quite simply, it's the sperm in males and the eggs in females. Question 2.4, what is the process when the gametes join together? Well, what do we call a sperm getting into an egg? It's called fertilization. The sperm fertilizes the egg and therefore it starts the new life. Question 2.5. Figure 1 shows cell division by meiosis to form gametes. Which two features in figure 1 show that this cell division is meiosis and not mitosis? So, we've been given meiosis here. We can see the cell, it doubles its DNA. It then splits once, and then it splits twice. And we can see what happens to the amount of DNA that from where it started, it's got half the amount. Because gametes, your sperm and an egg, have half the amount of DNA compared to a normal body cell, because obviously they're going to fuse with another sperm or egg to give a complete set of DNA. So we've said what makes it specific, what meiosis is, and we've labelled this diagram. Now, hopefully you should know about mitosis, which is when you have one cell and you make a genetically identical copy. So what happens is, in mitosis, you double the amount of DNA, it then splits and divides once, and you make two genetically identical cells from what you started with. So if anything, mitosis is a bit more simple than meiosis. So now we've got our content, we can look at the options. Which two features in figure 1 show it is meiosis, not mitosis? Tick 2 boxes. Well, a cell divides twice. Yep, that would definitely be it, because in meiosis it splits twice, but in mitosis it only splits once. The chromosomes are pulled apart in the new cells. Well, that's not specific to either one. Okay, so it's not that. The cytoplasm divides into new cells. Well, that happens in both, so it's not that. The DNA is copied. Again, that happens in both mitosis and meiosis, so it's not that. And the new cells have half the number of chromosomes, so that is a key difference as well. In meiosis, you've got half the amount of DNA. In mitosis, you've got double the same amount as what you started with. It's genetically identical. Okay, so moving on to question number 3 then. This question is about coordination in the human body. Figure 2 shows a sensory neuron, which is a nerve cell. You've got the skin, okay, and the label D. Then you've got this blob in the middle, and you've got the end of the nerve cell. Okay, so we can see it's quite long. Which label is the nucleus? Now, although this cell looks quite different compared to a standard animal or plant cell, we know a nucleus is generally the blob in the middle of it. So therefore, it would be A. What label is the receptor? Now, the receptor is the part of the cell, part of the body, that detects a stimulus, that detects a change. So generally, they are on the outside of the body. You know, our eyes, ears, nose, tongue, and also our skin. Okay, so our skin would be the receptor, so therefore it is D. Question 3.3. Figure 3 shows the nerve pathway when a person touches a sharp pin. So you've got a sharp pin, pricking someone's finger, it travels along a sensory neuron, and then it travels along a relay neuron, then it goes to the muscle. We've got to name structures A and B. Loads of people actually miss this question. Make sure you read everything on the page because it is tucked right at the bottom. Okay, so label part A, where we can see it's the gap between two neurons. Okay, the arrow is pointing to the gap between it. Now, the space between two neurons, two nerve cells, is called a synapse. They talk to each other by releasing neurotransmitter, which is just a chemical which diffuses across the synapse to activate the next neuron. Then it goes to the relay neuron, and then we've got to name this other type of neuron that goes to the effector, which is the muscle. So the 3 types of neuron are always sensory. It's always the first one that goes to your central nervous system. Then you've got your relay neurons in your spinal cord, and then you've got the motor neurons which carry it back to the effector to make something happen. So B would be the motor neuron. Question 3.4 then. When the finger touches a sharp pin, the muscle in the arm contracts to pull the arm away. What type of action is this? So this is an action where you're not thinking about it. It's automatic contraction. So it's not a conscious action, because that would involve you thinking about it. It's not a delayed action, because that would slow you down. It's a reflex action. It's a fast action to keep you safe. Question 3.5. Doctors tested people of different ages to time how long it took between touching a sharp pin and the arm muscle contracting. At each age, they tested 5 men and calculated a mean value for the time. It shows their results. So you've got the age 20, 40, 60, 80, and you've got the time for the muscle to contract, 18, 20, 23, 30. How much longer does it take for the muscle of an 80-year-old compared to a 20-year-old? Give your answer in seconds. Well, you can see from the table, at 80 years, it takes 30 milliseconds. And then at 20 age, it takes 18 milliseconds. So the difference is 12 milliseconds. Okay, now that is 12 milliseconds, so we need to divide it to give it into seconds. Now, because you're going from milliseconds to seconds, you would divide it by 1,000. So 12 divided by 1,000 is 0.012. Okay, divide it by 1,000 because you need to convert it into seconds. Question 3.6. Figure 4 shows the position of some of the glands which release hormones. You've got A, B, C and D. Which label on figure 4 shows the position of the pituitary gland? Now the pituitary gland is the one at the base of the brain, so it is A. If we just label all of them, pituitary is at the base of the brain, you've got your thyroid gland in your throat, on the top of the kidneys you've got your adrenal glands, and then D is pointing to our leaf-shaped organ used in digestion and hormones, which is the pancreas. Luteinizing hormone is a hormone released by the pituitary gland. What is the function or the job of LH? Tick one box. Now you should remember LH is a hormone involved in the female menstrual cycle. So therefore we'll look for the option that best fits that. So it's not to do with blood glucose, it's not to do with making sperm, it's not to do with the growth of muscles, it controls the release of an egg in the female menstrual cycle. Question 3.8. How does LH travel from the pituitary gland to the target organ? So this is basically asking, how do hormones get around the human body? And it's quite a simple answer. They travel round in the blood or the blood stream. Okay, either blood or bloodstream would get you the 1 mark. Question 3.9. Figure 5 shows the relative levels of sex hormones of the three young people over 30 days. One person is an 8-year-old girl, one is an 18-year-old boy, and the other is an 18-year-old girl. Which person is the 18-year-old boy? So if we look at the hormones they've told us about, you've got estrogen, progesterone and testosterone. Now we should know testosterone is the male hormone. So if we have a look at the graphs for testosterone, you can see that testosterone is low on person A and B, indicating they were probably girls, but it's much higher in person C. So therefore person C is the boy, and the reason, because they have high levels of testosterone. Question 4 then. A class of 8 students measured the population of water fleas living at the edge of a large pond. This is the method that each student used. They put water in a white tray. They took a pond net and scooped at the edge of the pond a few times. So as you're reading it, you should potentially be thinking of good things and bad things about their method. They emptied the pond net into the water in the tray. They counted the number of water fleas in the tray. Figure 6 shows a student working and how they're doing it. The students did not control some variables. Give 2 variables the students should have controlled to make this a valid method. So the key thing that I would have picked up from this method is they're not being specific at all, they're just saying scoop it a couple of times. So what should they have done? They should have scooped it the same number of times, and then they should also be really specific about where they are scooping it. So ensure it's the same distance from the edge. You could also say the same depth. And then you could also talk about the net itself. Okay, it has to be the same size net, same material, same size holes. Okay, any of them would get you the 2 marks. The students then used a different method to explain these results. Table 2 showed their results. So you've got this table here, you've got students going from A to H, and you've got the number of water fleas per per 1,000 centimetre cubed. You have to calculate the student's mean for the population of water fleas at the edge of the pond. Okay, so in order to do this, we are going to have to calculate the mean, which means add them all up and then divide by how many there are. So I can see if I add them all up and then divide by how many there are, so it would be our total. You would just plug it in your calculator to give you your total, and there are 8 pupils. The total divided by 8 gives us 64 water fleas per 1,000 centimetre cubed of water. What is the range of the student's results? Well, the range is the difference between the highest and lowest. So our highest is 122, our lowest is 19. So your range is 19 to 122, or you could actually just work out the difference between it, which is 103. Okay, so question 4.4, suggest one reason why such a wide range of results was found. Okay, so the reason why they may have found a big range of results could be just simply that the water fleas are not evenly spread around the pond. Okay, they probably live in little clusters, so they are not evenly spread across the pond. Okay, or it might be that more fleas live near the vegetation or the where their food actually is. So there is a big distribution of it. Question 4.5. The teacher then sampled the centre of the pond 8 times. So he's not doing the edge of the pond now. The teacher's doing the middle of it. He found his mean value was 12 water fleas per 1,000 centimetre cubed of water. What conclusion can you make about the distribution of water fleas in the pond? Use your answer from question 4.2. If you remember on 4.2, it said the mean of the water fleas at the edge of the pond was 64, while the mean in the centre is only 12. So what can we say about a conclusion? We can say there are more water fleas at the edge of a pond. Scientists counted some different invertebrates living in a pond between 2014 and 2016. It shows the results. So you've got bloodworms, freshwater shrimps, mayfly nymphs and water snails. Calculate the change in the number of bloodworms between 2014 and 2016. So have a look at the table. It goes from 13 to 48. So the population's gone up. So what is the change in it? Well, 48 minus 13 gives you 35. So it's the difference between it. Question 4.7. Calculate the number of shrimps in the pond in 2016 as a percentage of the number of shrimps in the pond in 2014. So we can see in 2016 there are 9. In 2014 there were 24. So it's gone down, and we need to know that percentage. So what you can do is 9 divided by 24, okay, times that by 100, and that gives you a percentage of 37.5%. You can round it to 38%, but you'd still get the mark if you left it at 37.5. Okay, so question 4.8 then. Invertebrate species found in a pond can be used as an indicator of the pollution level. Table 4 shows the species which can survive in different levels of pollution. So bloodworms can survive in all 3 pollution types. Freshwater shrimps can only survive in low and medium. Mayfly nymphs can only exist when there's low pollution levels, and water snails can live in all of them as well. What conclusion can you make about the change in the level of pollution in the pond between 2014 and 2016? Give a reason from your answer and use the data in tables 3 and 4. So if we have a look at what ones survive and what pollution levels, the biggest change is for mayfly nymphs. They only live in low water pollutions. They don't live in medium or high. So we could go back to our previous table and have a look what's happened to our mayfly nymphs. Well, there were loads in 2014, but there were none in 2016. So what does that tell us about the pollution in the pond? Where it tells us that the pollution must be going up because their number has gone down. So we could say, 1 mark, the pond is more polluted. Why? The number of mayfly nymphs has decreased. Question 4.9. Water pollution and global warming are 2 problems that have been caused by the rapid increase of the human population. Suggest 2 other problems caused by the rapid increase of human population. Now, there's quite a few answers to this because it's a suggest question, so you've just got to make sure you're putting some logical answers. So big issues that have come up because there are so many people about could be, obviously, there is more food shortages or water shortages. You would also obviously say there's more waste about because there are more people, which is putting more stuff into landfill. There's obviously more pollution that could cause acid rain. People need space to live, so therefore there's deforestation. Cutting down trees, so there's areas for people to go. And then you could also say there's extinction of species because we are using their land, their habitats for our resources. Another one you could say is natural resources are running out. So you just need 2 from that list to say the issues of human population. Question 5 then. Variation in an individual organisms can be caused by genes, the environment, or a combination. Figure 7 shows variations in a woman. What is the cause of each variation in table 5? Tick only 1 box in each row. So, what controls your eye colour? Is it your genes, the environment, or a combination? Well, your eye colour is just controlled by your genes. Okay, the environment has no effect on it. Light brown skin colour, so your skin colour is determined by your genes, but it also can be influenced by the environment and how much sun gets on it. So it would be both of them. Short hair is a style, okay? Therefore, it is determined by the environment. If it was saying hair colour, well, that could be potentially, if it's just natural, it would be your genes, but if someone's added colour to it and changed the colour, then that would be the environment. But just talking about the length of the hair, that is a style, so it is environment only. Question 5.2, the allele for blue eyes is recessive, therefore it is a little b. The allele for brown eyes is dominant, which is a big B, capital B. Remember, an allele is a version of a gene. Okay, so these genes both code for eye colour, but they're different versions of eye colour, giving different colours, therefore we called them alleles or alleles. A woman has blue eyes. What are the woman's alleles? Okay, so if our combinations are big B, well, she has 2 brown eyes then, so in that situation it would be brown. If she had one of each, she'd have a dominant brown and a recessive blue. The dominant is stronger, so that is shown. So she would still have brown eyes. So therefore she'd only have blue eyes if she had little b, little b. Question 5.3. The woman marries a man with the alleles big B, little b for eye colour. What colour eyes does the man have? Well, we've just worked it out here. Because he's got a dominant, he would have brown eyes. Complete the Punnett square for the diagram in figure 8 for this man and woman. A Punnett square is just another word for a genetic diagram. Hopefully you've seen these boxes before. So for woman, we know her genotype is little b, and then we combine them together. So big B, little b, big B, little b, little b, little b, little b, little b. What is the probability that a child of this man and woman will have brown eyes? So we can see we've got 4 potential outcomes. Each one is worth, because it's 1 out of 4, 25%. So you've got 25% of landing in each box. We've got 2 boxes that would give brown eyes, and we've got 2 that would give blue eyes. Okay, so therefore the percentage chance of this man and woman having a child with brown eyes would be 50%. Question 5.6 then. What is the scientific term used for the child's eye colour? So, we are actually now describing the feature that is shown. Well, the chromosome, that is where the DNA is stored, so it's not that. The condition, that's not that. The genotype is telling you what genes it is, like big B, big B, big B, little b, little b, little b. The actual description of what you physically see is called the phenotype. So the phenotype describes the actual colour, if it is brown or blue. What effect will a mutation have? Okay, so a mutation, if you remember, is a change in the DNA. Okay, it might be caused by radiation, but it could just happen naturally. Would a mutation almost certainly have no effect, definitely change in appearance, definitely be passed on to all children, probably cause a disease? Okay, now the key thing to know about mutations is that they happen all of the time, but normally they don't actually do much, or if anything. It's very rare that a mutation happens and it then causes a disease. So it's not going to probably cause a disease, it's not definitely going to be passed on, it's not going to definitely change in appearance. 99.9% of the time, it will almost have no effect. Question number 6 then. Many biotic and abiotic factors can affect the growth of plants. Are the factors in table 6 biotic or abiotic? Tick 1 for each factor. So we've got to know what these words mean to begin with. Well, biotic mean living factors, so anything that is alive. Abiotic are non-living factors. So diseases, well, they could be like living bacteria or pathogens, like fungi, so they would be a biotic factor. Herbivores, animals that eat plants, are living, so they would be biotic. Temperature is not alive, it's just a condition, so therefore it's abiotic, and same with the amount of water about. Those two would be abiotic. 2 students investigated the effect of light intensity on the distribution of small plants. The plants are growing under a tree in a park. The students made the following hypothesis. As you move outwards from the tree, there will be more plant growth. Explain why the students thought their hypothesis would be correct. So, if you've got more, if you're going away from the tree, think what is going to happen. If I've got my tree, okay, the sun is shining, okay, what's going to happen as I move further away? Well, as I move further away, there would be more light away from the tree. Then you can think about what plants are going to, or grass is going to do with that light. There would be more photosynthesis. for your second mark. And then because it is 3 marks, we would probably need to describe what photosynthesis does, which to make glucose to help the plant grow. Okay, so we have said what's happening, there's more light as you move away, for what process, and we're saying what photosynthesis does, which is to make glucose, which the plant can then use to grow even bigger. Question 6.3 then. The students use 2 pieces of equipment. Give the scientific name of each equipment. So a square frame measuring 0.5 by 0.5, so it's literally just a square, and we call this piece of equipment a quadrat. Hopefully you'll remember going out onto the school field, maybe counting the amount of daisies using some metal quadrat grids. What device would an electronic device used to measure light intensity be called? You may not have used this, but a logical answer is just quite simply a light meter, because it's measuring the amount of light. So this is the method the students use then. They fix one end of the tape measure to the base of a tree. They fix the other end of the tape measure 11 meters from the tree. At 0 meters, they put the square frame on the ground. They identify all the plant species growing inside the frame. They estimate and record the percentage cover of each species. They measure the light intensity inside the frame using the light meter. And where they put the square frame on the ground every 2 meters along the tape to 10 meters. They record the steps 4 to 6 in every frame. And you've got the diagram there that shows what they are doing. Okay, so they've got this metal grid and they're moving it along, and they would have put it down 5 times. Therefore, 6 times. So 0.5 times by 0.5 is 0.25. And they would have then put it down 6 times. 1 at 10 meters, 8, 6, 4, 2, and 0. Okay, so times it by 6, and that gives you 1.5 meters squared. Okay, so that is the total area that they sampled. Question 6.5 then. The whole investigation was done as quickly as possible on the same day. Suggest a reason why. Well, you'd want to do it quickly on the same day because weather conditions change. So it's probably to keep light intensity kept the same throughout the test. Question 6.6. Give one way the investigation could be improved. Now, the most obvious thing is when you've got a method like this, always look to see if they've done repeats. If they haven't done repeats, then that's a clear improvement. So they could repeat it around at different positions around the tree. They could repeat around other trees. They could count every 1 meter instead of every 2 meters. So it's only a 1 mark answer, so you only need 1 of them to get that point. You've got the table of results shown below then. So you've got the percentage of cover of each different species, grass, plantain, daisy and clover. You've got the distance in the tree from a meter. You've got the total percentage cover of plants going up, and then it tells you at the bottom the light intensity from the light meter. So as you go further away, you're getting more and more light. Which species in table 7 will only grow at high light intensity? So we know the highest light intensity is here. Which species only grows at high light intensity? Well, it's daisies, because they're not growing at this point when there's not enough light. So your answer would just be daisy. Okay, so question 6.8 then. What conclusion can be made about the relationship between light intensity and the total percentage cover of plants? Use data from table 7. So we're going to have to go back to table 7 and use data for 2 marks. But let's start it off. What is the relationship between light intensity and total percentage cover? Whereas light intensity is going up, what's happening to our percentage cover? It's going up. So that is our first bullet point. As light intensity goes up, so does percentage cover. But because we've got to use data, let's go back to the question and see if there's anything else we can spot. So light intensity is going up and up and up, okay? But we can see that at 175, you reach 100% cover. So actually what is happening is it doesn't matter how much more light you add, you can't go above 100%. Okay, so that would be your second point. Up until 175 light intensity units, where it reaches 100%. Question 6.9 then. Light intensity might not be the cause of this pattern of plant distribution. Suggest one factor that may cause these results. Okay, give a factor and then explain it. So, because light is not potentially increasing it anymore, there might be something else there that is potentially decreasing the amount of plants near the tree. So what could it be? Well, if the tree's there and it's big and it's covering the ground, there might be a lack of water hitting the ground. So therefore, the reason of it, okay, so the reason is the lack of water. The reason is the tree roots absorb all water, so no new plants can grow. And the same logic and the same reason could apply to a lack of minerals and ions in the soil. Because the tree is so big, it's taking all of the good stuff from the soil, so no new plants can grow. Right, last question then. It said, Pseudomonas bacteria can cause infections in hospital patients. Okay, a new strain of Pseudomonas bacteria has evolved. It can be only killed by one type of antibiotic called fluoroquinolone. Scientists want to prevent the new strain of Pseudomonas from spreading in the human population. Explain the advice doctors should give to prevent the spread of the new strain. So we are doctors and we were telling the doctors what to do to stop this new strain spreading. Now a question like this often throws students because you've got these big words, Pseudomonas, okay, and fluoroquinolone. These words though, they're not actually important. Effectively, it's just saying bacteria cause infections, there's a new one, it's killed by an antibiotic, you want to prevent this new bacteria from spreading, what should doctors do about it? Don't get thrown by these really complex scientific words. Try and strip back the question and think about what is it actually asking me. So, what could you do to stop this bacteria spreading if you were a doctor? Well, the first thing is you do not prescribe it for viral infections. Okay, remember antibiotics only work on bacteria. They do not kill viruses, so don't give it out for a common cold. And then secondly, you should only prescribe it for severe cases, okay, and not for mild infections. Why is that important? Well, because the human body can treat mild infections. It's always good for your immune system to keep active and kill any pathogens. Okay, if you do prescribe it, then the patient has to make sure that they finish it. Okay, so ensure the patient finishes the whole course of antibiotics. Okay, because if there are any left over, they may become resistant. Okay, so those are your kind of 3 key main reasons, and we've explained it for each one. Okay, so you say why you do it, and then you say a reason why. Other things that doctors may give advice to do is obviously things we've all been doing at the moment, which is washing hands in antibacterial gel or hand wash, just to kill any other bacteria that might be there. Okay, so that's the end of this paper then. Remember, the mark scheme and the paper is attached in the description, along with different videos if there are any specific topics for any questions you need to go over again. Okay, hope it was useful. Thank you.